is CORDIScovery. Hello and welcome to this episode of CORDIScovery with me, Abigail Acton. Did you know that when antelopes browse on an acacia's leaves, the tree will emit ethylene, which acts as an alarm signal to other acacias nearby? The gas can spread for 45 meters. Within half an hour, the trees that pick up the signal flush their leaves with very bitter tannins to make them less palatable to the antelopes. In high concentrations, the tannins can even be fatal. Most of us are focused on what we can see, touch, and feel or hear. We can hear one another. We know that some kind of communication is happening when a dog barks. But we are less aware of the interactions going on all around us that we don't perceive. So let's think again. Leafcutter ants diligently farming fungus, bumblebees picking up tiny amounts of electricity given off by flowers, all part of an invisible tapestry of interaction that is unfolding all around us. And perhaps we can work within that interconnecting web. Could the warning gases emitted by cereal crops under attack by pests lead to a more targeted approach to pesticide use, for example? Welcome to this episode of CORDIScovery, where we are celebrating the unperceived intricacies of the world with Daniel Robert, Professor of Bionanoscience at the School of Biological Sciences at the University of Bristol in the UK. Daniel explores how insects respond to weak electrostatic fields, a previously unsuspected form of interaction between plants and their pollinators. Hello, Daniel.
Hello, nice to be here.
Good to have you. Ted Turlings is based at the University of Neuchâtel in Switzerland, where he is Professor of Chemical Ecology. By unraveling how plants defend themselves against insect attacks and how specialized insects have evolved, Turling's team hopes to create novel, sustainable methods of pest control. Hello, Ted.
Hello, great to be here.
Nice to have you. Associate Professor of Ecology and Evolution at the University of Copenhagen in Denmark, Jonathan Schick is particularly interested in ants, their coexistence with other species, and how they survive in diverse environments impacted by climate change. Hi, Jonathan.
Hi, great to be here.
Nice to see you. Daniel, I'm going to turn to you first. The ElectroBee project investigated the first example of electroreception in a non-conductive medium that we have seen, the bumblebee's ability to detect and learn about the weak electric fields that arise when they approach a flower. So can you tell us a little bit more about what electroreception actually is, Daniel?
Indeed. Well, electroreception is the capacity of organisms to detect electric fields and or charges. So it had been studied for a good 60 years now in sharks and rays and usually aquatic animals in a conductive medium, which is water. So there it's quite well understood how it works. However, it had never been considered in a non-conductive environment such as the air, where, as we know, most of the species live, including an awful lot of arthropods. So we took on that little challenge to say, okay, if it exists in the water, could it exist in the air?
Fascinating, absolutely brilliant. What did you do actually to establish whether this was actually happening? Tell me a little bit more about the experiments you conducted in ElectroBee.
So we first realized that there was something going on electrically when we looked vaguely at YouTube videos first and then reproduced that in the lab, looking literally at pollen jumping from flowers to bees. And having a few students in the lab that were of a mixed breed, if you want, some people were physicists, some were biologists. We all look at it, and a physicist said, but look, the pollen doesn't drop randomly. It goes straight according to that nice little curve that you can see there on that video. There is an electric field. So from then on, we actually thought, okay, it may well be that because all matter is charged in the universe, it's not only on Earth and between bees and flowers, that we had to study the electrostatic charges taking place between bees and flowers. So we did experiments where we gave bees a choice, basically, go to a particular type of flower we put in the lab that was very artificial at the beginning, and one was actually with electricity and a bit of sugar, and the other one without electricity and without sugar. And we asked the question, can they learn? Can bees learn the difference between the two? And lo and behold, that was one of these moments of surprise that actually they could tell the difference.
Wonderful. And how did that manifest itself? I mean, first of all, how did you measure the amount of electrostatic being given off by the plants? And I think we're talking about the same thing that happens when we stand near a cathode ray television, if anyone can remember those from the distant past. I'm revealing my age a little bit here. But the hairs on your arms would stand on end, or a balloon at a kid's birthday party rubbing on your head. We're talking about that type of electric charge, aren't we, Daniel?
That's exactly that. These are very weak fields, yes.
Well, exactly. Weak fields and invisible. So tell me more about how you actually measured how much charge was being given off by the flowers. And also you say the bees learned. How did you see that actually happening? How did you test that they were learning?
So, one of the challenges was, of course, to make these fields visible or measurable. So they are small, but they are measurable. The problem was that there is no camera or microphone equivalent to measure electric fields. What you can measure, however, is the effect of a field that a field has on an object. So we shortcutted the whole argument by saying, okay, let's provide differences in electric fields to the bee and have that bee, by associative learning, learn whether there is a higher probability of them finding the source of sugar when there is an electric field to that particular flower. And we could measure by the behavior of the bee whether they could measure that field or not. And of course, in a control experiment, we canceled everything that we know was in that particular flight arena. We canceled all the electric fields, and we showed that bees that had learned to find the source of sugar with respect to the presence of a field, when the field was not there, they were unable to find the source of sugar, even though they knew they were learned bees. And that's the good thing about bees, is that you can teach them stuff and then basically test them, whether they remember, but whether they can still find the source of what they want, the sugar. So that's what we did, yeah.
Wonderful. And I understand that you at one point had them flying through hoops. Can you explain a little bit why you were making the bees fly through hoops?
Indeed, because we wanted to know quantitatively what sort of charge bees have. It had been reported in the 70s and 80s that bees were charged positively. And that also gave us a bit of a hint that indeed there would be an interaction electrostatically, like the balloon you were mentioning before. So one way to measure electricity or electrostatics without a contact is to produce a hoop or a ring made of copper. And we could also train bees to fly through hoops. We had to do that. That was much fun. And as the bee, as a charged object, flies through the hoop, it induces a little current. And measuring that current in picocoulombs, a fancy unit we like to use, we could actually make sure that we knew that the bee was providing a charge towards the flower. But then, of course, we also measured that the flower is reacting to that charge. And this is called technically mirror charging. But what happens there is that if a bee comes with, say, 5 charges towards the flower, that flower will summon -5 charges. So there is going to be a plus-minus game going on where the flower is not there passive, sitting quietly. It will actually play that electrostatic game with the bee.
Wow, so there is really direct interaction going on between the two of them. Excellent. Daniel, why? What's the benefit?
That was the key question, because we thought, okay, pollen jumps from a flower to a bee. This was known before, obviously. Aristotle saw that. I mean, there is the first, early reports on that. But for us, what was important is that when we saw the bees interacting with the flower was changing its electric status as a function of the visitation of the bee, which gave us the idea then that not only there was an interaction, but the bee knew about it. And that was a key question. Do bees know about the presence of these fields? And following from that, to cut the long story short, the idea was if the flower changes its electric status, it may well be that when a bee fly across a meadow, that not all flowers are equal electrostatically. And if flowers that have been visited by bees have a different electricity, a different level of electrostatics, that gives information to the bees, as we tested in the lab in the first place, that flower is more ripe with nectar or pollen, and that bees' foraging efforts, because time is precious for bees, are not really worth it. And that's what we could show, is that when amongst many flowers, if some have a different status, the bees readily measure that and try to avoid these flowers, but go back later.
That's fantastic. That's fantastic. And of course, also, I would imagine, you must have also considered other insects. I'm thinking, for example, what about night pollinators? that are working with senses beyond colour and vision, maybe sort of moths and stuff. There are the evening pollinators, night pollinators. Does that enable that process?
Indeed. So that was later research, and in the ElectroBee project, we broadened up the scope to see what other pollinators could do about this. Now, of course, I'm not going to say that all insects and all flowers do the same thing. We know that there is a vast diversity, but we could measure that some moths were electrostatically charged as well, whilst some others, like hawk moths, who fly very fast, who have a very high wing beat rate, actually do not charge up as much. So there's a whole different currency, so to speak. Some will have different tricks to their trade there, and but we think that in general, this is something that flowers need to do, is to keep the customership of their pollinators. And for that, they use good smell, they use good colors, good structures, and we would like to add to that they need to have also a very enticing electric field, as odd as it may seem at first.
No, it's just absolutely wonderful. It is totally a hidden world, isn't it? And massively complex. And are you seeing any other examples, Daniel? For example, predator-prey interactions rather than pollinator and pollen?
Yes, indeed. I mean, we focused on pollination because this is where we saw the phenomenon first and it came to our minds. But now we went a bit broader. And indeed, one of my students, Sam England, managed to record with this hoop system I was talking about before, the charge of predatory wasps, like the common Vespula, the common wasp, and also some wasps in Costa Rica, placing these hoops in front of wasps' nests. You have to be brave to do that. And then we could measure that there was a charge. But then he also measured caterpillars. And we saw that caterpillars are more likely to engage into a defensive behavior and stay in that defensive behavior when the electric equivalent of the flight of a wasp, if you want, because they have an electric field. So we could record that with electrodes. And if we play back, in the absence of wasps, this electric signal through the air, which is not conductive to the caterpillars, they will actually stay in their defensive mode and or start flailing, start to drop off of the leaves. So there are all sorts of dramatic or sometimes less dramatic behaviors. But caterpillars, we think now, are aware of the presence of their predators through the electric field that arises between themselves and the predator. Whether it's generalizable to all caterpillars and all wasps, I can't tell you that. But for those that we've observed, at least 3 examples, including in the tropics, we can say that now there is a new channel between prey and predators that is being opened by that.
That's just wonderful. I think I love doing these podcasts, and I'm reminded very regularly of a vague feeling of arrogance. You know, we always seem to think that we know everything that's going on, and then we open a page and suddenly there's a whole world of stuff that we haven't previously considered very closely at all. Thank you so much. That was a fascinating explanation. Does anyone have any questions for Daniel? Yes, please, Ted, go ahead.
Yeah, so Daniel, you were explaining to us that this allows, this electrical charge from the flowers allows these wasps to determine which flowers carry the most pollen or nectar.
Bumblebees.
Yeah, the bumblebees. Doesn't this open the possibility for the flowers to cheat, that they would actually manipulate their charge?
I'm just going to step in because I think that's a wonderful question. And actually, Daniel, I was wondering too. I mean, for example, can wasps mask that? I mean, do the creatures and the plants play with this? Because, I mean, it must have been around for millennia.
I think so. I think this is an excellent question because we know that as soon as there are signals between different organisms, they can be as different as a plant and a wasp or a bee or caterpillar. There will be some room for exaggeration of traits, but also for concealing the truth. We think that flowers that will benefit from what is called in the trade flower constancy, where the pollen needs to go back to the flower of the same species, with serial pollinators that actually stick with the same type of flower day by day, that will not pay off to lie. However, there are other flowers that are more ephemeral, that open, need maybe even to be a bit shaken or be self-pollinated, and or the pollen has actually a very much higher specificity and can afford to be somewhere else but then be picked up again. There are all sorts of possibilities. So what Ted is asking is that indeed some flowers may not react the same way. We haven't found an example yet, sadly, but the idea would be that there perhaps the electricity is just used to attract and mimic what good attractive flowers are. Just like other flowers will have a good color and a good scent, but they will have a good electrical taste, if you want.
Fabulous. Thank you so much. That was excellent, Daniel. Really appreciate that. I'm going to turn to you, Ted, actually. I'm beginning to feel a bit sorry for caterpillars, and the listener will now find out why. So your project, AgriSense, set out to develop a novel detection device that can interpret when a plant is giving off odours, indicating that they are under attack. Ted, what drew you to the role of volatile chemicals in biotic interactions, please?
Yeah, this goes back a long time when I started my PhD, which I did at the University of Florida. And I was given the task to find out how parasitic wasps, which are similar to the wasp that Danielle was describing, they prey on caterpillars, how they find these caterpillars and then they lay their eggs in there. And after some behavioral assays, I discovered that they actually use plant odors to find caterpillars that are feeding on a plant. And when studying that in more detail, we discovered that the plants are responding to caterpillar attack by producing a very specific odor that then attracts these wasps. And this odor, the fascinating part about it is that it's not just the mechanical damage that these caterpillars do, but there's something in the oral secretion, spit if you want, of the caterpillars that is recognized by the plants, and then they start synthesizing new volatile compounds that they then emit. And this blend of volatiles is attracting these wasps.
That's just stunning. Really, really. Can you tell us a little bit more about this? Because I believe that you conducted experiments where the plants weren't necessarily being damaged by the caterpillar, but the wasp was still coming. Tell me a little bit more about the experiments you conducted.
Yeah, then we actually, to find out that these oral secretions were important in this context, we mechanically damaged plants ourselves and found that if you don't add caterpillar spit to the damaged areas, the volt emissions were much, much lower, and several of these compounds that they normally emit in response to caterpillar attack were not emitted. So that's how we were able to prove the importance of this oral secretion, and we developed a bioassay where we even could incubate plants, and we worked with maize plants that we cut at the stem and just put them in a solution with or without caterpillar spit, and then we saw a tremendous difference in the volatiles emitted by the plants, even if the leaves were not damaged after several hours. Yeah.
How intriguing is that? Absolutely. And so, this is absolutely fascinating, but do you have some sense of an application that could then grow out of this knowledge that we have?
Yeah, that was exactly this AgriSense project, looking into that. So the idea there was that if the wasps are able to recognize plants under caterpillar attack, would we also be able to detect that? And if the odor is specific enough, then you could maybe even detect what insect is feeding on the plant on the basis of the odor emitted by the plant. And there are subtle differences in what different caterpillar species, but also other insects, and even pathogens like bacteria and fungi that attack plants, induce the plants to start producing specific odors. And
these odors that the plants produce, are they systematically in order to protect themselves, like, for example, with regards to the predatory wasps, or are they simply emitted as distress signals without an end goal, as it were, by the plant? Do they each have a target?
Yeah, what the original function was is a little bit hard to determine, but we also, as you mentioned at the beginning of this podcast, is that there is also this option of plants interacting with each other through these odors, and that's one of the possible functions. I hate to use the word communication because it might be just eavesdropping. Let's name it that way. A neighboring plant can pick up that odor, realize that there's a caterpillar or another
a challenge of some sort.
Challenge, yeah, and then they start preparing themselves for that possible attack. And they can do that also by increasing actually their responses in terms of producing defense compounds, or also producing these odors to attract the natural enemies of the herbivores. Mm. And we have done quite some research on that, and we have found out that pathogens and insects induce considerably different odors, and that we actually can pick up on that. And we now have tested several sensors that can detect these odors in real time. And the eventual goal would then be to have that install those sensors on a rover, for instance, that goes through a field of maize plants and smell individual plants and then give immediate real-time feedback to farmers where there is a problem. And you could even imagine that the same rover would then apply something to kill that pest. And logical thing would be pesticides. But we're also looking at other options here.
Such as what, Ted?
Well, one of the possibilities that we're really looking into are nematodes, tiny little worms that kill insects within 1 or 2 days. They're parasites of insects, and we have developed now a formulation of that we can inject into the whorl of a maize plant, the center of a maize plant, with nematodes, and it actually works.
Do they, though? Do they? Do they work? Yeah, that's good, because I must admit, I have entered the world of the nematode. I was plagued by slugs in my garden this summer, something chronic. I applied nematodes 2 or 3 times, and slugs really seem to not mind at all. So I hope that your nematodes are more effective than the ones I was using.
Actually, we have worked with the slug nematodes as well, and they kill much, much slower. And that could actually take weeks to months before they kill the slug. So in the case of caterpillars, they get killed within 2 days. All of them. With just 1 or 2 nematodes, you kill them right away.
Wow. Okay, so there we go, fellow gardeners. Keep applying the slug nematodes. It won't work all at once. Thank you very much. Super. Do we have any questions for Ted? That was excellently explained. Thank you. Yes, please, Daniel.
It's fascinating. It's really great because it really unveils what Ted is doing there, these hidden aspects of if it's communication or if it's dropping. I think at the end, it doesn't matter at all. There are so many different complexities. I was wondering, Ted, whether, because we know there are lots of caterpillars that become toxic as they choose to eat plants to actually deter predators. Is there a relationship between the toxicity, the non-palatability of these caterpillars and the way they can secrete these very substances that become their demise as well? Is there a double game there that actually some caterpillars have managed to avoid and still be deterrent, but also deterrent to all what is negative to them through a same process of toxification?
Yeah, specialized insects that are able to actually sequester the defense compounds of plants. And these specialists have found ways to detoxify these compounds themselves there. And we just recently published a paper on a beetle in that case, where the larvae feed on the roots. And the adults feed on the leaves. And those adults are able to sequester the defense compounds from different plants, different toxins. And that was not known yet, that they can actually sequester multiple different defense compounds. And they transfer those compounds to their eggs that are then protected against the predators. And it's another fascinating world that is a bit different from the communication aspect of it.
Well, it's relevant if we think of it as interactions rather than communication.
Yeah, exactly.
It's interconnection and interaction. Actually, frankly, a bit mind-blowing. I think it's wonderful. Absolutely brilliant. Thank you very much for that question and for that answer. I'm going to turn to Jonathan. Jonathan, the Elevate project looked at the interaction between leafcutter ants and the fungus they farm. All right, so we're still talking here about this kind of notion of symbiosis, really. Leafcutter acts as farmers. I mean, this is not something that immediately comes to mind. Jonathan, tell us more, please.
So yes, if you've ever had the chance to visit a rainforest in the New World from, Panama to Argentina, you'll often see these massive highways with thousands and thousands of ants just foraging. And it's kind of one of the most iconic scenes from a rainforest. And what they're actually doing is climbing to the tops of the trees and cutting all sorts of leaf fragments from hundreds of different species, and then taking them back to the nest where they disappear underground. And it's what happens underground that's really interesting to me. And it turns out, like you said, these ants are real farmers. They vertically transmit a crop just like human farmers from generation to generation. They protect it from all sorts of pests and pathogens. They create an optimal growth environment underground, and it is their food source. It converts plant material that they can't eat into an optimized nutritional reward that the ants depend on for their livelihood.
Okay, I did wonder. So thank you very much for answering that. So when you say creating a food source, that the ants don't eat the fungus itself, I presume, do they? Or do they eat what the fungus produces?
So they eat the fungus. The fungus produces these very specialized, domesticated fruits, like an apple on an apple tree,
right,
that the ants eat.
Okay, but obviously then it doesn't damage the fungus at core, because otherwise they'd be eating their own kind of necessary crop.
No, you're right. It's a very unusual thing for a fungus to do, to provide food for another organism. But it turns out because this is an obligate system, these different species depend on each other. They can't live without each other. That it makes sense evolutionarily that the fungus just gives some nutrients to the ant farmers.
Right, without then getting damaged itself, really literally like apples on trees then, in fact. Okay, exactly. Fascinating. So what were you particularly interested in within the Elevate project? What did you consider closely there?
So I've always been fascinated by how ant colonies work. Each individual ant is pretty dumb. I spent many hours watching them in the rainforest on trash bags on the leaf litter, just watching them forage. And if you make a footprint in the leaf litter, some ants of some species will just wander aimlessly for hours, in circles. But somehow these farming ants are able to manage these incredibly complex, sophisticated farming systems. And so I was wondering how they could do that. And I specifically focused on nutrition and communication with the ants, with the fungus, because human farmers know exactly what blends of nutrients are best for their crops. My question at the heart of my grant was, how do the ants know which blends of nutrients make their fungus crop grow the best? And how can they actually detect that? And can they make complex nutritional decisions to optimize productivity of their food crop?
And I noticed also that you were interested in sort of different climatic impacts as well, so that situations would vary depending on the context.
Yeah, I think that's true. These ants range all the way from southern New Jersey, different species, not necessarily leaf-cutting ants, but it's actually a quite diverse lineage of ants that are all farmers, that are all related to each other, all the way to Argentina. So they range from rainforest to desert to grassland. So they have to be able to grow their crop in really diverse habitats. And they've also, unlike humans who have been farming for about 10,000 years, these ants have been sustainable farmers for about 60 million years.
Oh, wow.
So how have they managed to thrive across all this climate change was also a key interest of mine.
Yeah, sure. And so how did you go about studying the leafcutter ants? I know, for example, you went to Panama. Can you tell us about some of the experiments you conducted in the Panamanian rainforest? Must have been quite a challenging environment.
Yeah, so we work near the Panama Canal at the Smithsonian Tropical Research Institute, and it's quite a beautiful forest. There's these huge container ships navigating the Panama Canal, but we're just with our head in the leaf litter looking for ants. But one of the things we did, like I mentioned, we actually stole bits of leaf material from foraging leafcutter ants as they walked by, and we let the ants go on their way. And then we collected all this plant material and we analyzed it for all sorts of nutrients and all sorts of secondary metabolites like toxins that the plants might produce. And in this way, the ants could tell us what nutritional blends they're actually foraging for, how they're kind of what I call navigating a nutritional landscape of hundreds of plant species, and whether the nutrients that they're foraging for in the field match the nutrients that we can actually measure in the lab that maximize the performance of the fungal cultivar in a petri dish.
And did you then try growing the fungus in the labs with the nutrients that the ants had been collecting and also without? I guess you had a control.
Yeah.
So what did you get? What were the results?
So it's complicated because nutrition is always complicated.
Yes.
But we could show that things like protein can quickly become toxic to the fungus when you grow it in a petri dish. And yet somehow the plant material that they're foraging in the field has more protein than you would predict would be optimal. But we were able to show that there's all sorts of secondary nutritional interactions that actually make the protein palatable to the fungus and allow it to thrive despite getting a bit too much protein in the field. And not only that, but the ants can really detect how much protein that they're depositing on the fungus, which is quite interesting.
Absolutely.
And we don't exactly know how they do that yet, but they can. And that's part of the research we're currently working on.
And they modulate, I guess, how much they supply to stay within parameters that are secure for the fungus. Fabulous. Well, this is what the ants get out of it. You can see all the effort that goes into it and why they get their ant equivalent of apples from the apple tree, as it were. What does the fungus get out of all of this?
Well, it gets protection and it gets to be vertically transmitted. So when a new queen flies away from her colony of birth to start a new colony, she takes a little bit of fungus in her mouth. Then she mates with several males. The males die. They're just flying sperm missiles. She falls to the ground, loses her wings, digs a hole, and then spits out that little bit of fungus from her colony of birth and starts to take care of it. And then she lays eggs from her mating and starts to produce workers, and the colony grows into a new colony. So the fungus doesn't have to produce mushrooms, doesn't have to invest in reproduction. It gets free food by the ants.
Dispersed.
And it gets dispersed to the next generation.
Yeah, brilliant. Absolutely wonderful. And you mentioned something about the ants protecting it. I think you were touching on the possibility when we previously talked, Jonathan, of the fungus perhaps producing a compound that could make the ants more aggressive in their protection of it?
Yes, so there was work done by another tried to investigate a bit, that shows that the fungus metabolizes certain lipids, certain fatty acids that are slightly different than what's in the plant material, and it sequesters those or accumulates them in their fungus tissue. When the ants kind of sense that, they can become a little more aggressive and protect the fungus garden. We're currently looking into kind of, as Ted kind of was interested in volatile signals, we're more interested in kind of the kind of long chain carbon, or these larger molecules that kind of coat the fungus garden, because we think we need more kind of really targeted signaling from the fungus. Too much protein in this spot, change to carbohydrates, or there's really bad plant chemicals here, take it away, or there's a pathogen here, remove it from the fungus garden. So I think volatiles would be more too broad of a signal.
Right, something more specific, like a tap on the shoulder.
Exactly.
Yeah, I think that's wonderful. Absolutely brilliant. I'm loving this. This is excellent. Thank you very much, Jonathan. Does anyone have any questions for Jonathan? Yes, please, Daniel.
Jonathan, this is wonderful, wonderful stuff. And I was wondering, because I've seen these in the field as well, and I find them fascinating as well. Well, I'm not working on them directly. This is amazing. Now, fungal spores are everywhere, and I can see these ants going to a tree, and they forage on that tree, and they must be bringing some fungal spores with the leaves. Is there any evidence that they select the leaves or they clean the leaves before to prevent some sort of an import of all sorts of pathogens, fungal pathogens? Because fungi are not very nice to each other. How do they protect their apple tree if in that case?
Yeah, that's a really good question. So there's many different solutions that the ants have, and it varies across species. But it has been shown that a lot of plant leaves have things called endophytic fungi, or fungi that kind of live inside the leaves kind of quietly their whole lives. And some of those fungi can produce compounds that the fungus doesn't like inside the nest. And it has been shown by other groups that the ants can select against leaves that have specific bad fungi living inside the leaves. But the ants produce all sorts of chemicals to protect against different pathogens, like fungal and bacterial pathogens inside the fungus garden, all sorts of chemicals. They also have bacteria that live on their bodies that produce antifungal and antibacterial compounds that can also protect against this really bad fungus called Escovopsis that's really bad for the fungus garden. And so it's a very kind of complex web, as we might say, symbiosis of different organisms protecting and defending.
And trying to get around those protections and defenses as well.
Yeah. And it's quite interesting. If you dig a hole underground, like, half a meter, you'll find a chamber, and the soil environment is so dirty, but the fungus garden is just clean, separated from the fungus and a plant root or something, and the ants just are constantly interacting with it and cleaning it.
Very assiduous farmer. Thank you. Thank you, all of you. That couldn't have been more interesting. It was really, really wonderful to talk with you. Thank you for spending some time with us.
Thank you very much.
Thank you.
Thank you, everyone. Bye-bye.
Bye.
Goodbye. If you've enjoyed today's fascinating insight into a hidden world that perhaps none of us have previously thought much about, follow us on Spotify and Apple Podcasts, and check out the podcast homepage on the cordis.europa.eu website for other episodes that are equally interesting. Subscribe to make sure the hottest research in EU-funded science isn't passing you by. And if you end up chatting to somebody about podcasts you're enjoying, why not name drop CORDIScovery? We've talked about MRI scans of baboons and how they inform our understanding of the development of language, and how it could be possible to collect samples from tiny asteroids millions of kilometers away. In our previous episodes, there's bound to be something that will tweak your curiosity. Visit the Cordis website, which has articles and interviews that explore the results of research being conducted in a very broad range of domains and subjects. From structural color to structural dynamics, there's something there for you. So come and check out the research that's revealing what makes our world tick. We're always happy to hear from you. Drop us a line. editorial@cordis.europa.eu. Until next time.